Morning, everybody. Can everyone hear me okay? Excellent. Good morning. Welcome to everybody in person. Really good to see everybody here. Okay, so welcome to the CAS Bioassay conference. What I'm going to do for the first 10, 15 minutes is go through some introductory remarks and some housekeeping items, and then we'll have our plenary presentation by Helena. All right, so on the screen is your internet password. I think everybody might be aware of that from the flyers. But that's an internet password. A couple of important things to note. Please don't use your cell phones. Put it on mute. Obviously, that can be quite distracting to the folks around you, but also to the speaker. And photos and videos with your phones are not allowed. If the slides are deemed acceptable by the speakers, they will be available through the CAS site. So what is CAS? And for folks that might be new to CAS, this is a kind of an important organization, and folks that may be aware of CAS is a good reminder. CAS is an agile, nonprofit scientific organization whose strength is in bringing together professionals from industry, academia, and regulatory agencies to solve scientific and technical problems in order to advance the development of pharmaceuticals. One of the things I want to mention to you guys and the audience is what the committee really likes to do with this conference is to make it as interactive as possible. We'll have panel discussions after the end of the speaking sessions. We highly encourage people to ask questions, and there's always a robust conversation during those sessions. So write down your questions that you have on your notepads, and we'll, feel free to then ask those questions during the panel discussion. CAS is committed to inclusion, to diversity, equity, and inclusion in all aspects of what we do. Diverse perspective, drive innovation thinking, and better solutions that advance our mission of regulatory capacity building, knowledge sharing, global access. We advance together in the direction of dignity, respect, scientific truth, and the betterment of humanity. CAS is an organization that is really built on knowledge sharing. The purpose is to discuss best practices and trends in the field of pharmaceutical development and regulation. As you guys know, we're all in our own independent companies. We do our own things. But one of the important pieces that you learn from here is benchmarking. What are other companies doing? Are they using the same practices as you? And that's one of the things that's really valuable coming out of this conference. Feel free to have those conversations around benchmarking and shared learning with other colleagues and speakers of interest. CAS sponsors various types of scientific conferences. We are obviously in the bioassay, and we are obviously the center of the universe, right, guys? But CAS also sponsors CE, mass spec, and other high order structure and other meetings. But again, we're the center of the universe. CAS has a global reach, and as you can see, we've got participation from global geographies. As you can see, from significantly from North America, but also from Europe, Asia, and other parts of the globe. We have folks from various disciplines that come to our conferences, right? So it's not simply R&D scientists, but we have a significant amount of folks from regulatory affairs. As you will see, we've got an active participation, heavy participation from regulatory folks. Certainly FDA is in town, but we have participation from European regulatory agencies and other countries as well. We have folks from QA, QC, engineering backgrounds, marketing, business development, et cetera. So it's quite a broad organization from participation perspective. Here's the most important slide. Here's the committee members that worked tirelessly throughout the year to put the programs together. And one of the things I'll mention, and we'll mention towards the end of this conference, a couple of days, is your valued, as participants, your valued opinion on talks and topics that you want to hear about. So, take a note of who these committee members are and approach them as you go through the conference and you feel like, hey, you know what? there are some topics that we'd like to hear in future conferences. Ping them, email them, etc. And none of this would be possible without the tireless work of a CAS staff. You might have met Julie outside, but there is a host of people that support this organization and make this possible. So a real robust thank you to you guys. CAS has program partners listed here, as well as symposium exhibitors that you guys might have already seen being propped up in the hallway, in the hall there. So visit them, talk to them. They're always coming up with new technologies, new things to check out. So drop by and talk to those guys. Our media partners are Bioprocess and IPQ. And as a reminder, feel free, if you're comfortable wearing masks appropriately, feel free to do so. There are sanitation stations as well. And last but not least, make sure you can connect with the CAS app with the CAS community as well. Okay, we have a new member networking break from 9:30 to 10 in the Lakeside Ballroom, which is around the corner on the other side of this hotel. This is a great opportunity to just kind of network, hang out, kind of learn a little bit more about what folks are doing, what this organization does over the years and has done over the years. And so it's a great way to kind of find out more and essentially just network with people. All right, so I think we are now ready for our keynote speaker, a superstar in our own right. Can we get her slide on? Yeah, thank you. So our speaker is Helen Madden. She's a Senior Director of CM&C Regulatory Affairs at Bluebird. But prior to that, she spent 19 years at Biogen, both in the regulatory area as well as in the analytical sciences development. So she's got a wealth of experience in the bioassay space, particularly from a regulatory perspective. So she's a fantastic speaker. I should mention that Helena used to be a stalwart member of this committee, and so we've known her for a number of years and are excited to have her back. She's got a lot of wisdom, a lot of knowledge, and she's happy to share it with us. Helena holds a PhD in biochemistry from Boston University and a postdoc from Harvard Medical School. So we really are excited to hear what she has to say. I should mention really quickly, she'll be also on the panel discussion after the first session. So write your questions down, and then she can take those questions during that panel discussion after the first session. Okay, thank you.
Good morning. Good morning to everyone. It is so fantastic to be here. Thank you for inviting me. I hope by the end of my presentation that I can convince you guys that bioassays are the superstars of drug development. I think Bhavin already has that in his mind. Okay, so this is my disclosure statement. I'm at Bluebird Bio, and I own stock in the company, so these are my views and not the views of the company. So I'm not sure if everybody remembers me, but this is my caricature from our 2019 conference, which was our last face-to-face meeting. I can't believe that we missed 3 years in there, and now we're back. So it's so awesome to be here face-to-face, and I'm seeing so many familiar faces around the room. It's awesome. So let me tell you a little bit about the beginning of this conference, because I think it's kind of interesting, and it also shows you what you guys could do. So in the early days of monoclonal antibody development, I was asked to bring a bioassay development group into analytical development. And I complained to my boss. I said, there are really no places to talk to other people in other companies, to talk to regulators, to learn about bioassays. And he said to me, Helena, don't complain to me, make one. So he told me to write a one-page proposal, who should come to this meeting, what you should talk about, some logistics. And I did that, and I gave the one-page proposal to somebody involved in the organization of the CAS meeting, and here we are today. And many of the same people who helped organize the meeting are also here today. So we decided that we need to talk about scientific approaches for the execution, for the development, for all aspects of bioassays, and also regulatory strategies, regulatory expectations. And this is not a static subject matter, because the world evolves, things change, and we develop new biotherapeutics. The objective and the secret sauce for this meeting has always been conversation. We're all humming the same tune. We want to get safe and efficacious drugs to patients who need them. I also want to thank the organizing committee for agreeing to move the keynote speaker to the first day. Typically, you guys have had the keynote speaker on the last day, but I really appreciate the opportunity to set the stage for this meeting. Okay, so this is what I want to talk about today. I want to talk about bioassay basics. I think we have been, as a scientific community, we've been spurring off like a lot of startup companies and a lot of new small thinkings. So I think we need to come back to some basics and really learn some things again. We're going to talk about bioassay challenges and also what can bioassays tell us, and then some words from my experience. Okay, so some bioassay basics. Well, what is a bioassay? A bioassay shows that a drug candidate does something and has an activity in a living system. Our early ancestors, they chewed a berry, they smoked a leaf, they drank tea, or they made paste from a plant part, and they watched for some beneficial or medicinal effect. In 1984, Paul Ehrlich, he was working on animal bioassays, and he started the idea of standardizing in order to quantitate. He created standard preparations against which he could test unknowns, and he defined a unit of activity based on the activity of the specific mass of his standard preparation. Today, bioassays play a role in many industries and in everyday life. I like to judge science fairs, and at these science fairs, I've seen so many cool bioassays. We should take a lesson. But those are usually the winners. Bioassays are important in the food industry, especially beer and wine. I don't know why that comes to me. And also I note that they put my water in a wine glass. So here we go. One of the most famous bioassays is the canary in the coal mine. And we're here to talk about bioassays and biotech. So bioassays can be used to measure an in vivo or an in vitro response in a drug discovery or a research setting. We can use bioassays to determine product activity in animal models of disease, to select drug candidates, drug targets, and production clones, to show PK, bioavailability, and to evaluate immunogenicity. Results of bioassays influence design of clinical trials and can demonstrate understanding of a therapeutic biological pathway or mechanism of action. We use bioassays to characterize and control a drug substance process, to test a drug in a QC lab for assurance of biological function and molecule stability. In gene therapy, we use bioassays to show the function of a transgene, and when you're developing biosimilars, bioassays are really something that you rely on to show the similarity between your product or your candidate biosimilar and the originator product. We also need to use bioassays when you make a product for a new indication or if your current product has a new mechanism of action. So we talked about how do we use bioassays, But why do we need bioassays? Well, through all phases of clinical development, all health authorities have expectations and requirements for assurance of identity, purity, potency, and stability of biotherapeutic drug substances and drug products. According to ICH Q6B specifications, assessment of the biological properties constitutes an equally essential step in establishing a complete characterization profile. A valid biological assay to measure the biological activity should be provided by the manufacturer. A relevant, validated potency assay should be part of the specifications for a biological drug substance or drug product. And some examples include animal-based bioassays, cell culture-based bioassays, biochemical assays, or some other procedures like ligand and receptor binding assays. So I mentioned the word potency. What are some definitions of potency? According to ICH Q6B, potency is the quantitative measure of biological activity based on the attribute of the product, which is linked to the relevant biological properties. The assay demonstrating the biological activity should be based on the intended biological effect, which should ideally be related to the clinical response. According to U.S. Code of Federal Regulation, the word potency is interpreted to mean the specific ability or capacity of the product as indicated by appropriate lab tests or by adequately controlled clinical data to affect a given results. Tests for potency shall consist of either in vitro or in vivo tests, or both, which have been specifically designed for each product so as to indicate potency as in the definition above. So when I first started working on monoclonal antibody development, I studied these definitions really intently. The highlighting is my highlighting. It does not come from the ICH Q6B. But I studied these definitions in order to convince upper management to provide the resources that we needed to develop a potency assay and bioassays for every single product. That's a really important concept, and I think that for some of the complex products, we still need to do some convincing. I've been very impressed by health authorities because they've been putting out guidance, potency assay guidance for complex products. For example, cell and gene therapy or cancer drugs or combination of drugs and such. And I put links to those guidance and also ICH Q6B and a bunch of other things in a couple reference slides at the end of this presentation. So what's the bottom line? A well-controlled potency test shows a relationship to the underlying mechanism of disease and is critical for all phases of drug development. A well-controlled potency test provides a quantitative measure of product bioactivity and function and is required for drug substance and drug product release testing. A potency test evaluates product stability and shelf life and provides assurance of consistent product manufacturer. We also use potency tests to assess comparability of products following manufacturing changes. Okay, so let's switch gears a little bit and talk about some bioassay challenges. If this stuff was easy, we wouldn't be here, right? Okay. Potency assays and bioassays are specifically designed for each product. They should represent the mechanism of action for a specific product. So I want to think a minute about the definition of what we're talking about. Maybe delivery or binding of your drug to a cell or uptake of your drug to a cell. It could be the pivotal function, but you need to ask yourself, is there more? When you define the clinical or primary MOA, ask yourself, the product binds, and then what happens? Does it block something? Does it activate something? Does it express something? Does it kill something? And ask yourself, is there a secondary MOA? For example, for monoclonal antibodies, FC effector function can be a secondary MOA. A product can bind to a specific Fc gamma receptor on an effector or immune cell, and then result in antibody-dependent cellular cytotoxicity, or ADCC, or antibody-dependent cellular phagocytosis, or maybe the molecule binds to receptor C1q and activates complement. And just one thing to keep in mind, Fc effector function can also be a primary MOA. So the idea is that you need to work with your research group to verify that the in vitro format of the bioassay that you're developing is relevant to their understanding of the MOA. And you might need multiple methods to define potency. If there's more than one mechanism of action, you need more than one potency assay. If you cannot eliminate effector function, you need to monitor effector function. If the clinical MOA is unknown, having multiple potency assays may be advisable because you're really not sure what's going on. And we need bioassays to control manufacturing process and also for drug substance and drug product lot release. Many times these are the same assays but run in a different format. For lot release, assay performance is key. We want to run more replicates and fewer samples per run. And you'll also need to have bioassays for molecule characterization. Many times these methods are not a good fit for a QC lab, but they provide very, very useful information and they can be used for specific situations. So we know complex products are difficult to characterize. Well, that's the reason why potency is a critical parameter. A potency test or combination of tests can model the interactions of complex pathways in an in vitro environment. And remember that potency tests show that a consistently manufactured drug product is administered through all phases of clinical development. A phase-based or incremental approach for development of bioassays is acceptable and advised, except for the situation of biosimilar development. In early phase, you want to think about target binding and the methods need to be qualified. In late phase, you'll need to have a cell-based functional assay, and that needs to be validated by pivotal trial and the phase 3 GMP campaign. But you should think about developing that as early as possible because that's the most work. And that method is also something that you'll learn a lot from. If you use different assays through the course of development, and maybe you have changed vendors and you're using a vendor assay, but if your assay changes, you need to bridge that information. So what should a gold standard potency assay look like? The deliberate drug design should be considered. The mechanism of action should be reflected. The method should be accurate, should be reproducible, should be robust, and should be stability indicating. In addition, the method should be practical. The format should be simple. Cell lines should be available and stable. Critical reagents, for example, commercial assays, antibodies and such, should be available and stable. You should evaluate lot-to-lot variability of critical reagents, and you need to make sure that appropriate reference standards are available. Instrumentation and equipment should be dependable and needs to be qualified or validated, and clear methods for analyses and results reporting should be defined. If you're running a bioassay in more than one site or more than one lab, I really recommend having one control chart for all of your testing sites. This is a way to catch problems early. And make sure that you retain samples for evaluation of future method changes because we all know that things change. So when I first started working on bioassays, the big problem was method variability. Well, that was then and now is now. We really have learned by a lot of work from folks like you about how to control variability, so we really can measure potency now. So let's talk about a few things, about a few ways to reduce variability. Well, one is according to the US Pharmacopoeia, Chapter 1032, because of the inherent variability in biological test systems, an absolute measure of potency is more variable than a measure of activity relative to a standard. Assuming that the standard and the test materials are biologically similar, Statistical similarity should be present, and the test sample can be expected to behave like a concentration or dilution of the standard. So we determined relative potency. And there are lots of ways lots of tests we can do to talk about relative potency, tests for parallelism, equivalence, curve similarity. There have been lots of great presentations at this conference. To reduce variability, use platform technologies such as reporter gene assays. Use platform instrumentation, and the equipment should be routinely calibrated. Standardize cell culture systems. Use easy-to-culture cells. Create or buy ready-to-use cells, which are banks or aliquots of cells, which are frozen at the same passage, the same concentration. And the idea is that these can be taken from a freezer, and the concentration in the cell passage has been studied and is optimum for the best recovery upon thaw. The use of ready-to-use cells and also consistent culture conditions really standardizes assay performance from time to time, from assay execution to assay execution, and results in convenience, flexibility, and really helps with method transfer. One thing to keep in mind when thinking about reduction of variability is to allow for sufficient assay development and performance optimization time. Don't do less. Evaluate and optimize every method, including data analysis. Most folks use a design of experiments approach and maybe repeat the evaluations to confirm conclusions. Study and optimize plate layout and think about plate uniformity. To reduce variability, you might want to shorten incubation times, and you might need to amplify the signal as a result. One thing that I learned along the way is if your potency assay depends upon sample concentration, don't rely on the sample concentration that comes to you from another group. Verify the sample concentration in your lab at the time of execution of the assay. And we all know that automation of method unit operations like liquid handling, pipetting, and such really decreases variability because robots can pipette the same all the time, so they can reduce analyst error. They can pipette viscous solutions. They can reduce analyst fatigue. There are just a lot of great advantages for automation. But one thing to keep in mind is that automating a bad assay, it's still a bad assay. So make sure that you don't forget to give enough time to develop your assay and optimize the performance. You also need to develop meaningful system suitability criteria and sample suitability criteria and give time for your analysts to train. They need to practice. They also need to be in an environment free from distraction. And one thing that I like to give thought to is method transfers. Method transfers should be well designed. The transfer of a bioassay to another site or another lab or another country, that can be really tricky. One way to decrease variability is to co-validate the methods in the transferring lab with the transferring lab in the receiving group. The transferring lab and the receiving labs really need to work together to successfully transfer a bioassay. So many times, critical reagents are overlooked. What are the critical reagents for bioassays? And bioassays have a lot of critical reagents. Cell lines, antibodies, labeled antibodies, recombinant proteins, serum, assay controls, reference standards, commercial assays. There's a lot involved. And you need to think about, how are you going to source the critical reagents? Are you going to make them in-house? Is one lab going to make them for everybody, all your sites, or are you going to buy them commercially? And then you need to think about characterizing the critical reagents because you want to make sure that you get or make the same critical reagent each time. So you need to characterize and qualify them. And think about the handling of critical reagents and each one of these critical reagents. For example, will you aliquot them? Will you freeze them? How will you thaw them? Will you reuse them after you thaw them? How will you store them? What is the stability, expiry dating, retesting? There's a lot to think about and a lot to remember to document. Changing critical reagents could impact a potency assay. And when you do change reagents, you need to use predefined procedures and also predefined acceptance criteria for new reagent lots. Okay, a couple of words about reference standard. This is one of the main critical reagents. According to ICH Q6B, at the time of submission, the manufacturer should have established appropriately characterized in-house primary reference material prepared from lots representative of production and clinical materials. In-house working reference materials used in the testing of production lots should be calibrated against the primary reference material. You need to do this for all modalities, even the complex products. The potency of bioassays is typically expressed as percentage of the potency of the reference standard, right? Percent relative potency. And a link needs to be maintained between the potency of the reference standard used during clinical trial development time, in characterization studies, and in commercial release and stability studies, and that goes post-approval as well. You need to think about qualification of reference standard potency and use the appropriate number of replicates when you qualify a reference standard potency. Acceptance criteria for reference standard qualification should not include manufacturing variability. Don't use potency data from multiple lots to set acceptance criteria, and do use multiple replicates of a single lot. In 2019, Susan Kirshner gave a really great presentation at this conference. I put a link to that below. She shows some case studies, and there's some great ones on reference standards, so I recommend you have a look at that. Okay. So what can bioassays tell us? This is actually my favorite part, so I hope I didn't get carried away teaching you a little bit about that. But I really like when we can increase our product knowledge by bringing together people who understand bioassays, people who understand analytical chemistry, people who understand biophysics, and put all our heads together to solve a common problem. So how does your product work? We need to continuously learn from clinical trial results and also continuously learn from analytical characterization of your product. Conducting clinical trials for the evaluation of every single product variant or every manufacturing change, that's just not very practical. It takes a lot of time to run clinical trials, it's really expensive, and they're really not all that sensitive to small changes. So use bioassays. Bioassays can be used to correlate the bioactivity to the structure and to the mechanism of action, because remember that the bioassay is the link back to the clinical response. Relationships can be derived between bioactivity and protein structure. Changes in higher order structure can impact activity, bioactivity. Post-translational modifications can also impact bioactivity. For example, phosphorylation, changes in phosphorylation can influence cell cycle, cell growth, apoptosis, and cell cycle signaling. Glycosylation is important to think about. The absence or the presence or the type of a carbohydrate can influence bioactivity. Product variants, for example, oxidized variants or clipped variants, they can result in a loss or a gain of activity. So characterize your product to understand what product attributes contribute to the biological effects. You never know, measurement of a post-translational modification may be more sensitive than a method that measures bioactivity. So is a potency assay always a biological test? Let's go back to ICH Q6B again. A relevant, validated potency assay should be part of the specification for a biological substance or drug product. When an appropriate potency test is used for the drug substance, an alternative method, it could be physicochemical or biological, may suffice for quantitative assessment of the drug product. However, the rationale for such a choice should be provided. You need to justify your rationale using supportive scientific data, and that's where characterization comes in. You could use a cell-free assay that reflects the MOA to assess potency, and some examples might be receptor binding on a microtiter plate rather than on a cell. Receptor phosphorylation using a cell membrane prep and not on a cell, or direct binding to a specific ligand. And that brings up the question, is a potency assay always a biological assay? Well, we know the answer to that, but you're not going to get away with not developing a cell-based functional assay, because correlation to a cell-based bioassay is an expectation. The cell-based assay is linked to the clinical effect, as we said before, and you need to do a comparison between your cell-free assay and your cell-based assay, and ask yourself many questions. Is the cell-free assay better? Is it less variable? Is it easier to transfer to QC? Is it easier to procure the critical reagents that you need for the cell-free assay? And you need to show this using data. If you do propose to use your cell-free assay as your potency test, think about proposing to use your cell-based assay for comparability, for validation, for qualification of a new reference standard. So surrogate analytical assays may be used to demonstrate potency if the following are shown: a solid understanding of product bioactivity and other critical product quality attributes, correlation to relevant product-specific bioassay results, and you need to use qualified or validated assays for these studies. Results from multiple lots of appropriate test articles and also patient samples are required. And these test articles need to be, for example, active species, inactive species, degraded species. Remember that correlations are powerful friends. The amount and types of correlation data needed to justify your approach and to develop your scientific rationale depends on product complexity. The more complex the product, the more data you need. Some people want to do the opposite, say, oh, my product's too complex, I can't generate any data. Okay. So let's talk a little bit more about determining the relationship between structure and function. There have been a lot of technology advances in the analytical chemistry side, especially mass spec. There's a lot of mass spec methods that really have given us the power to understand things and to deeply characterize our molecules. We know that structure is necessary for bioactivity, but data from structural assays alone, they're not enough to assure molecule potency. So structure-activity relationship studies involve perturbing the structure and then investigating, does this structural change impact bioactivity? Bioassays can inform about a change in product chemistry or product structure, but only if that chemical or structural component plays a role in bioactivity. And you need to consider the overall impact of your intentional structural change. Did any unintended changes occur that can confound your the thinking about your results? Demonstrating linkages between the structure of biotherapeutic and its function increases product knowledge. Use that product knowledge to your advantage when coming up with your scientific rationale for your potency assay strategies. Orthogonal methods that measure physical, chemical, and/or structural components of a product can serve as surrogate measures of potency, but they don't necessarily replace potency assays. A lot of work is needed to build up your scientific rationale, but we learn a lot along the way. So can physical chemical methods replace traditional bioassays? The answer is yes. A lot of work has been done on monoclonal antibodies, and there are a lot of literature references, especially literature references that show the impact of carbohydrates or N-glycans on bioactivity, on safety, and on efficacy. This figure shows a stick figure of a monoclonal antibody, and you can see that on asparagine 297, an N-glycan. The red triangle stands for fucose. The blue square stands for N-acetylglucosamine. The green circle stands for mannose. The yellow circle stands for galactose. And many labs have studied these N-glycans and disturbed the structure or added or taken off these certain sugar molecules. And there have been some conclusions that can be reached. For example, an increase in galactose results in an increase in complement-dependent cytotoxicity. A decrease in fucose results in an increase in Fc gamma R3A binding and antibody-dependent cellular cytotoxicity or killing. High mannose and no fucose also leads to an increase in Fc gamma R3A binding and ADCC activity. So we know that production of monoclonal antibodies and also Fc fusion proteins in mammalian cell culture cells results in a heterogeneous N-glycan pattern. While these N-glycans can affect the conformation or the shape of the antibody because they're charged. And in that way, they modulate interactions with Fc receptors on the immune cells and the impact effector function. So for monoclonal antibodies, N-glycan composition may be a critical quality attribute, and you might need to monitor this to assure potency and during product and process consistency. So let's think about some factors that are important for successful structure-activity relationship studies. You need to develop strong partnerships between your bioassay specialist, your analytical chemist, your biophysicist, and you need to think about resources. It takes a lot more time and people than you think to do an SAR study. Sample generation is one of the key parts. You need to create a library of variants on your molecule, such as a monoclonal antibody. Maybe you want to take off all the glycans or add a lot or add a mid amount of glycans. And you do this by expressing the monoclonal antibody in certain cell lines that don't express, for example, fucosyl transferase, an enzyme that adds fucose, or you use enzymes to clip off or enzyme inhibitors to stop the clipping off of certain sugar molecules. And then you need to think about chromatography enrichment and preparative chromatography separation of all of these different variants and enrichment of each glycan type. Then you need to characterize each variant. When you're making these changes, did you cause some aggregation? So you need to do size exclusion chromatography. Are there fragmented forms? Maybe capillary electrophoresis can be used to show that. And then mass spectrometry is really the method that's used to determine glycan structure in most labs. And mass spec can also pick up other modifications like oxidation or deamidation. And you need to also think about, when creating your library of variants, are these variants stable? Once you create mixtures containing various amounts of N-glycan variants, now it's time to think about evaluating bioactivity of each variant in your potency assay matrix. The figure in the square is a figure that Florian Seimer presented last year. I was very impressed by his presentation. In this figure, he shows the complexity of the assays versus the robustness and the sensitivity and the precision of this potency assay matrix. In the top left-hand square, functional cellular assays such as ADCC, ADCP, CDC, apoptosis, using cell lines or primary cell lines, measure binding and effector function at once. He did find that the use of primary cells leads to a lot of variability. Functional cellular assays using cell lines and some kind of surrogate readouts measure binding and also signaling. Cellular binding assays such as FACS, FRET-based assays or cell-based ELISAs measure binding, and also assess conformation. The readouts from these assays are very sensitive. And then cell-free binding assays measure binding and reduces complexity. So hopefully you can see that bioassays are a critical tool to show how monoclonal antibody structure influences bioactivity. Excuse me. And these principles can be applied to other modalities. When we start thinking about complex products, I'm really confident that it'll take us some time, but we really will understand how antibody cocktails, bispecific ABs, drug conjugates, gene therapies, and cell therapies work. So potency is a key parameter for complex products because there's a lot of unknowns and because they're difficult to characterize. You need to provide the rationale for your bioassay application strategy. Think about your overall procedure. What bioassays are you using, for what purpose, and why? What analytical tests are performed, and how do they complement or strengthen bioassay results? Combinations of all product quality attributes matter, but bioassays are the superstar. So I hope you all know to listen to your bioassay. If your bioassay won't work, maybe you're not understanding the mechanism of action. Maybe you're not measuring the whole mechanism of action. So if your bioassay is difficult to qualify or to transfer to QC, difficult to validate, maybe something's going on and you need to listen to your bioassay. It's telling you something if you only listen. Remember that drug design and also your indications may evolve over time, and you need to think about, did you change your bioassay if you need to? Okay. I will say 45 minutes is a long time to talk. I practiced my talk the other day, and I got tired in the middle. But I'm almost done. So a few words of, I would say wisdom, but the more words of things that I learned from my experience. Start potency assay development as early as possible. Work with your research lab. You know that you'll need to develop a cell-based bioassay. Don't wait until phase 3 to start that. Do it earlier. Understand the product mechanism of action and reflect that as closely as you can in your potency assay. Develop multiple measures of potency and document assay performance and suitability. I found that you really can't predict, you just don't know what will work. So developing more than one measure of potency is a good way to go. Excuse me. And also, you never know when you need to talk to regulators. So you really want to write down what worked and what didn't work so you're prepared for that. Potency assay development should be continuous throughout product development, and you should use a product life cycle approach. Think ahead, though. You know you need to validate. You know you need to do comparability studies. So think ahead from the beginning and get yourself prepared for that. Make sure that you measure potency in stability studies. Test multiple bioassay reagent lots, multiple instruments, and multiple analysts as much as you can. If you don't measure method robustness and you're running an assay that can only be run by one person or is not very rugged, it's not really that useful to you. Understand what product attributes contribute to the biological function. Evaluate the contribution of post-translational modifications and also structural changes. Talk to regulators about potency strategies, reference standards, any challenging issues, and listen to the regulators. So I hope I've convinced you that bioassays are the superstar methods. Please keep the conversations going between industry and regulators. And just a slide to acknowledge a few people. One, I definitely want to acknowledge the Bioassay Scientific Organization Committee. They are so dedicated, and I'm so pleased to see that so many people have been on since the beginning, since 2009. I especially want to thank Hannah, Denise, Katrin, and Evangelos. Those were the regulators that have been with us from the beginning. I want to thank them for the things that they taught us and for listening to us. I also want to thank Bob, Bruce, and Karen. They were really fundamental in setting up the first conferences. That's an absolute thing, and they put up with me. I definitely want to thank Svetlana Bergelson and the Bioassay Development Group at Biogen. That's the place where I learned about the power of bioassays. And I want to thank Leslie Wilder, Ben Matzma, and the Reg CMC flock at Bluebird Bio. I'm on some new adventures with them, and I'm learning a lot about bioassays for cell and gene therapies. So thank you guys, and a shout out to everybody who even thinks about bioassays. So. Thank you. I just want to point out that my last two slides are full of references. So if you want, to learn some things, go for it. Thanks, Bobby.
All right. Thank you, Helena, for a fantastic presentation. I'm sure she spurned a lot of thoughts and questions. Write them down, jot them down. As I'd mentioned, she will be on the panel discussion after our first session, which is the nuts and bolts of bioassay session. So right now, we're going to take a little networking break till 10 o'clock. So be back in this room by 10 o'clock. The new members, there's a special networking session for you guys because you're so special. And so we'll see you guys at the Lakeside Ballroom. And for everybody else, please be back here at 10 o'clock. Thank you.